everyone. My name is Shih-Wei Fang, and I'm a postdoc here in ICCP, working with Excel. And the title of my talk is A Climate-Driven Human Genetic Bottleneck in Africa 900,000 years ago. And here are the collaborators that in ICCP and also in Europe. And let's start. Okay. So I think a lot of people heard about this paper, who et al. In 2023, and they used modern human gene information, and then they try to see whether what's the effective population size over the past million years. And they found that there is a huge bottleneck, genetic bottleneck exists around the 900 Ka or 0.9 million years ago. And strikingly, and they found that this genetic loss is causing about like the effective population size is only fewer than 1,280 individuals, and then immediately bounce back to 27,000 individuals. And in their paper, they suggest that this population bottleneck between this 930 to 813 Ka, it's most likely because of the climate change. And also coincidentally, and there's a paper coming out post-Vika et al. 2022, and they suggest that at this same timing, the 0.9 million years ago, there's a chromosome 2 fusion exists in our human DNA. So next, what is the 0.9 million MA event? So as you heard from the last talk, we know that there is a really stable climate state before the mid-Pleistocene transition. And then after that, we have a stronger variability. And then in the 0.9 million MA event, it's around the MIS 22. And there's a large glacial increase at that time. And also, this timing is also the mid-Pleistocene transition. And as we heard previously, there's a huge difference before and after. And so that the hominins in Africa at this timing, it's facing a really strong and cool cooling around the 0.9 MA event. And then after that, they are facing a more variable climate environment. So it means that after the strong events, they are even more they are harder to survive because of the variable climate environment. So how are we studying this? We use the agent-based model. So I'm really lucky. So yesterday we heard a lot about the agent-based model. So the agent-based model is trying to simulate some simple interactions between individuals. So in our model, we simulate simple activities, including mating, death, birth, survival, movement, and also we include the mtDNA, the mitochondrial DNA, in our individuals. And the way we represent the African hominins, we use the habitat suitability model that also introduced in the previous talk. But here we use a little bit different ways. We use the African fossil, including habilis, Homo agaster, Homo heidelbergensis, and we use a 40 records running window in order to try to simulate the change over time, the habitat change over time for the African hominins. And last, to represent the past climate change, we use the net primary production, so food availability change over space and time in our agent-based model. So here you can see the MPP change. And then let's just look at the agent-based model and the results. So we start the simulation from 3 different places, the north, the south, and the east, where they have the records, where we have the fossils. And here in the top left, there's a year, although it's a bit small, but you can see when is the timing. So let's start. So after we started, you can see the 3 regions is having their own pattern. And there is some population in Central Africa, and it moves up and down with the ITCZ. And you can see now around the 900 Ka, the extinction in the central and north and the south. And there are some left over in, or not, survivals in the southern part. And then you can see the Eastern Africa is always really populated. So let's look at it again. Yeah, so you can see here, it's moving up and down with the ITCZ. And then they will extinct first around 1400 Ka. And then after that, there is a extinction in the north. You can see around 1000 Ka. And after that, around the 0.9 MA event, the southern part extinct. And now you can see at some events, the southern part will rebound back and then extinct again. So let's see. You can see the top panel shows the temperature change. We can see why we have those extinctions. It's because the temperature is going down all the way. And then around the 0.9 event, 0.9 MA event. And then we have 3 extinctions in the Central Africa, and then in the Northern Africa, and then in the southern part. So although here you can see we compare between before and after the 0.9 event, there's only like 20% of drop for the total populations. But because we use the mtDNA, the mitochondrial DNA, to trace the DNA change of our population. And the reason we use the mitochondrial DNA is because it's only tracked for the females. So it's a bit easy and computational less. So then if we look at the effective population size and the genetic diversity, we can see that although there's only a 20% drop in the total population, there's a 72% drop in genetic diversity during the 0.9 MA event. The reason why we have this huge drop is because the genetic diversity is highly depends on the population structure, but not the numbers of the population. So if you remember, in the southern part, the large population extinct, and only small, tiny individuals are staying there. So most of the genetic information is included in the eastern part. So that's why there's a really strong genetic the diversity drop. And then if we look at the following part, we have several spikes that representing the recoveries and the extinction in the southern Africa that you see in a previous movie. And we also try to assimilate with 10 more simulations, and then see how there's a difference, or is there something changes. So you can see the top left panel is about the Eastern Africa population. You can see the population is really stable. Like there's not many difference within the 10 ensemble members. And the lower panel shows like how many ensemble members have the survival there. So in gray means that all survives. And in red here means that only less than 1,000 people survive. So you can see in the Northern Africa, the extinct timing is a little bit different, but they all extinct before the 0.9 MA event. And for the Southern Africa, you can see that there's one simulation that entirely extinct. So only the Eastern Africa population is left. And then if we look at the genetic diversity, you can see that genetic diversity have a larger variability compared to the population. This is because we have some stochastic in our mutation, so that's why there is the larger variability. And next, we try to find which climate variable is the most important to trigger this 0.9 MA event. So we do the EOF or PCA analysis for separately for temperature, MPP, and precipitation. We can see the pattern. The temperature has the largest variability in the northern part, and the MPP has a large variability in both northern and southern part, and the precipitation is mostly around the Central Africa. So if we look at the PC and plotting with the diversity, we can see that they all vary pretty well with the diversity. You can see here. And then after the 0.9 MA events, they're kind of not really varied together, but they all have this strong drop at this timing. The reason why they are so vary together, it's because that these three modes, you can see here is EOF1, EOF1, and EOF2. These three modes are related to the CO2 forcing in the climate model. So you can see here, before the 0.9 MA event, you can see the CO2 variability and the diversity are changing like really well. And then after that, it's not that same, but then you can see these these three events is also vary with the CO2 change. So it means that the large-scale climate shifts can cause change in population structure and then change the genetic diversity. And next, we use another way. We try to keep constant for these three variables separately and calculate the habitat suitability. It means that since it's constant, it's not having that drop anymore. It shouldn't have the genetic bottleneck exist. So we see that for the NPP has the largest contribution to the bottleneck because you can see there's the largest numbers for the NPP simulations. And then the next one is the surface temperature. Since both of them are highly related to the CO2 variability, that's why they are really important. But if we see for the precipitation, you can see the bottleneck still exists. Okay, so next, we try to include the cultural development into our model. So in the page in Parallel 2024 paper, they provide the counts of procedural units. So what's that? It means that how many procedure you need to make the stones or make the instrument at that site or for those hominins. So you can see that the unit is quite flat before the 0.9 event, and after that it's going up. And so then we try to mimic this change of the cultural development. So we change the Kmax in our model, which is the maximum number of individual over the grid box. So it means that if it's cultural developed, so they can have more survival within the grid box. So you can see for the total population, after the 0.9 events, then it's just linearly going up. And then, but if we look at the effective population size or the genetic diversity, we can see that it's not really going linear go up directly. There's a pump around 100 Ka. This is because that at this timing there's a climate event that we see previously. There's a CO2 increase, and then cause the population to increase in the south, and then they are not dying anymore and just keep increasing after this timing. So our result is a bit different from the Hu et al. Because since we add the the cultural change. In their paper, they have a constant individuals, so that's really different. And then last, if we sum over the last period, last 100,000 years, we can see that these regions are connected. So the cultural development helps the interbreeding of within the regions. And if we only simulate from the eastern or from the southern part of the Africa, we found that the southern part can migrate to the eastern Africa, but the eastern Africa population cannot migrate to the south. So it means that the climate event has a direction, and then this also control the easiness or how difficult they can propagate to the other region. And if we being really bold, we increase the Kmax to triple. And then we can see the population can migrate even out to do the transcontinental dispersal to the Eurasia. You can see here is the timing that they disperse out the Africa. Of course, this is more like a experimental that's showing that increase of the cultural improvement can help the dispersal. Yeah. So that's it, the take-home message. Maybe you can just see by yourself. Okay, thank you.